Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Cell and Gene Therapy Clinical Trials, How to Successfully Operationalize a Trial of Prolonged Duration. My name is Samaya and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Allucents who developed the content for this presentation. Allucents has a depth of knowledge and a wealth of regulatory and clinical trial experience that encompasses the use of autologous dendritic cells, stem cells, and gene therapy for the potential treatment of a variety of diseases, including oncology indications, infectious diseases, degenerative joint diseases, neurodegenerative diseases, and various immunodeficiency disorders. With more than 30 years of experience in over 60 countries, Allucents' individualized partnership approach provides experience-driven insights and expertise to assist its clients in successfully navigating the complexities of delivering novel treatments to patients. Now, it's my pleasure to introduce our speakers for today's event, and our first speaker is Desmond Cabrera. As Vice President of Project Management, Desmond Cabrera is a seasoned professional with over 20 years of experience in leading cross-functional teams to deliver complex projects on time and within budget. He has a proven track record of success in managing large-scale projects across multiple therapeutic areas, including oncology, cardiovascular, CNS, and is a global ops lead for Allucents' cell and gene therapy portfolio. With extensive knowledge of project management methodologies and tools, Desmond is able to drive efficiency and productivity throughout the project lifecycle. He's committed to delivering high-quality results that exceed stakeholder expectations and drive business growth. And our next speaker is Joyce Moore. Joyce Moore brings over 20 years of industry expertise spanning multiple disciplines and a diverse range of therapeutic areas. For the past 17 years, Joyce has focused on patient engagement, recruitment, and retention. She is passionate about widening participation in clinical research and developing and deploying technology approaches to engage with both sites and patients, including decentralized approaches. In her current position as Global Head of Patient Engagement at Allucents, she is part of the clinical trial organization's leadership team. She is responsible for leading, managing, and overseeing patient recruitment activities and Allucents' decentralized trial offering known as Patient Direct Trials. And our next speaker is Marita Krushkoff-Osterberg. Over her 15+ years of experience within regulatory affairs and steady startup, Marita Krushkoff-Osterberg has gained extensive knowledge on how to successfully navigate the complex and integrate clinical study startup process. She has overseen the project startup phase of several global trials across a variety of indications and phases and has overseen submissions in 30+ countries. She is currently serving as manager for a team of EU CTR specialists and led the coordination of the first EU CTR application done by Allucents in early 2022. And finally, our last speaker is Francisca Sampson. Francisca Sampson is an experienced director of data management with a proven track record in leading and overseeing complex data management and clinical research projects within pharma and CRO. With over 30 years of industry expertise and 20 plus years within clinical data management, she has been involved in process optimization and team leadership. With her in-depth understanding of data processes, coupled with hands-on experience and an open mindset for new and creative approaches, she brings valuable insights into efficient data collection approaches that ultimately support analysis and reporting. So now, without further ado, I'd like to pass control to our speaker. So Desmond, whenever you're ready, you can go ahead and get started.
Thank you, Samaya. And I just wanted to take a second to say thank you to X Talks for giving us the opportunity today to bring something, that is from an operational standpoint that we see here at Allucent and really work to help our sponsors and move their therapies forward. How do you operationalize? What are some things that you want to really address? And appreciate the time today. And for everyone else, thank you so much for joining today. It's really nice to be able to speak to the therapies and the landscape because it is so cutting edge. And I really do get enjoyment out of seeing how these companies are progressing and how therapies are progressing, because ultimately it's going to be a plus for patients. So for today, we're going to go through a really quick overview of the cell and gene therapy landscape, what we would consider, pillars of success or the right level of questions as sponsors are bringing their trials to first in human usually, and then going through phase 2 and phase 3. What are the things to think about, prepare for, especially when you're looking in the EU from a regulatory perspective, and then what does startup look like, logistics, some data management strategies, and ultimately at the end of the day, we want to ensure our patients are engaged and are having a good experience. That's going to take us to what I would love to understand first is a polling question is for those in the audience, and I'll pass this back to Samaya for a second, and she'll run the overall poll. What is your current experience in cell and gene therapy?
Thank you, Desmond. And yes, that does bring us to our first poll question. So audience members, you should be seeing that poll question on your screens now, and you can go ahead and select the option that suits you best. And all you have to do is just click on that option. So that question is, what is your current experience in CGT? Your options are, my company organization is running multiple CGTs, or we're starting our first CGTs, we are moving into CGT, no actual trial identified, or no experience yet. So go ahead and click on the option that suits you best. So audience members, I'll give you a couple more seconds to answer that. And once again, that question is, what is your current experience in CGT? Okay, so I see that most of you have voted. I'll go ahead and close the poll and share the results now. The results are as follows. 56% of audience members voted for the first option. That's my company organization is running multiple CGTs. No audience members voted for option 2 and option 3. And then 44% of audience members voted for option 4. So that's no experience yet. Thank you everyone for participating. Back to you, Desmond.
Awesome. Great results too. It's always interesting to understand the audience and kind of where we are. Going to come back to the screen. Let me know. Can everyone see it? Good. All right. So really brief, and this is apparently for the 44% that don't have a ton of experience, and I'll just kind of put this out there. So cell and gene therapies are really changing the dynamic, and it's revolutionary in the potential for treating various diseases. We're operationalizing the trials with such a duration and complexity. It's a significant task. There's a lot of things that you have to think about that may be very unique to the cell and gene therapy trial, specifically in the autologous or the allogeneic therapies, where for an autologous, you're looking at using patients' cells to then modify and then infuse back into the particular patient, and that could really be a curative measure. And I think that's the term that I've heard most when you talk to KOLs and keynote speakers. It's the ability that it's not just a treatment, it's really potentially a cure. And as we continue to see the landscape grow, rare disease and genetic disorders, there really is a big window for opportunity for us. And I think that's really, really special for patients who, are dealing with those particular disorders. There's innovative studies at all levels of the clinical trial stage. And the CROs, they really sit in a unique position to ensure that these complex trials have the right level of experience, because obviously they're seeing it from several different sponsors probably at the same time, different type of technologies and different types of pathways, and it's really interesting. But you have to leverage that experience and leverage that knowledge to help all your sponsors across the board. And then, there's also the concept of it has to be collaborative. And I think, the term collaborative sometimes is thrown all over the place, but in cell and gene therapy, from an operational standpoint, the CROs, the sponsors, and the additional vendors, which include central laboratories, shipping components, logistics, which we'll talk about a little bit later, they're all so important to the stakeholders of the study, and then ultimately the stakeholder is that patient. So it's really nice to kind of see that relationship grow and how you can actually work to bring it to a place that makes sense. And then, we'll go from there. Just a quick chart, and this is sourced from the cell and gene therapy dashboard from Sightline in quarter 2023 in October. The number of phase 1 development, it's increased 7% since the second quarter in 2023. 58 trials were initiated for gene therapies, with trials for non-oncology increasing about 6%. And I talked a little bit about that before, that we continue to see gene therapy starting to take on more and more of the space, and it's because there's a progression and people are starting to understand the technology. But CAR Ts will continue to dominate the pipeline right now in the autologous setting or in the allogeneic setting, depending. And what the charts here show you is genetically modified cell therapy breakdown between the CAR Ts, and you can see that most of them sit 47% or so. The TCR-Ts are about 11%, and then your NKs are about 6%. And then when you look at the overall CAR T breakdown, it predominantly sits oncology. But although the non-oncology, I can say, is also starting to grow, because there's various programs out there that are starting to really look at different types of diseases and mechanisms. So really quickly, that's just a quick landscape of what it looks like. And I really do think that it's worth going to the ASGCT for cell and gene therapy to kind of get a real good feeling of where things sit. But for this particular conversation, what we really want to achieve is, what are some things to look at from an operational perspective? How do we prepare for it? So when a sponsor comes to us, or we start looking at trials that are going to first in human, and you're ramping that clinical development, there's so much time spent on the clinical development from the sponsor side to even get to this point. And now you're at that place where you're going into the trial. What are some questions, or what are some things that you have to consider right off the bat? So right here, what I think are some important pieces are, what is the current status of your manufacturing? Are there constraints in product development? Will those constraints impact your overall enrollment? How will you be presenting? Because obviously a lot of our sponsors have stakeholders that are looking for additional funding, things of that nature, and there's corporate goals. It starts with the right level of manufacturing, because it isn't as simple as just getting an additional lot of drug that we can keep at a pharmacy. These cells, this product, it's real life, it's people's cells, so it's not something that it's easily stored. Do we need to consider specialized vendors? I talked about this a little bit before, but there's a ton of logistics that are associated to a cell and gene therapy trial. That includes the manufacturing, that includes the shippers. So do you have the right central lab analyzing the samples? Do you have chain of custody, chain of command, chain of quality in place, so that way as cells are progressing through the chain, that you know exactly where they are, and that you have the right vendors in place that have the experience in cell and gene therapy. And then regulatory is evolving, and today specifically we're going to look a little bit at what the EU has kind of put in place from a regulatory perspective, and how it's impacted overall submissions for cell and gene therapy programs, and what it's done to particular timelines, things of that nature. But how do we achieve success when we start looking at strategies? And then once you get through the regulatory and you look at the countries that you're in, what is the right site? And that's a term that I've used for several years, and you could use it across the board on any trial, but really specifically, you have to be very smart about the type of sites that you need to bring in. And that's a level of experience that they have to have, the right level of PI experience plus staff experience. It's extremely important. And that flows into the overall data. It's a critical piece to the trial. And then how do we build that to really manage cell and gene therapy trials, like the data flow, and Francesca will go into it a little bit, talk about like what that looks like from the first 2 weeks of a subject signing ICF, or maybe 6 weeks through, long-term follow-up. And then patient engagement ultimately, and this is the most important piece a lot of the time, is we want to make sure we keep the patients engaged, but what are the significant challenges that we see with a therapy that has, mandated follow-up periods that can be, longer than someone owns a personal cell phone. So it's something that really we have to take into consideration. And how do we actually ease that burden? So when you look at the challenge for delivery, there's really 4 pieces to it that I think make a lot of sense. And we're going to go into that today for you. So we talked about regulatory a little bit. What is this country-specific regulatory? What's the expertise in that particular country? And understanding accelerated timelines, and understanding what you need to go through for the approvals, what cell and gene therapies might need a couple additional hurdles, timing, things of that nature. Then you have the overall site journey. It's outside the norm. It has to be a collaborative partnership, even at the site. There's a cell and gene lab most times. There's oncologists who are more familiar with the experience of autologous programs, because it's there's various products already out there. And when you start looking outside of the oncologists and other PIs that might be in other indications, how do you work that relationship to make sure that they are working as a good mechanical machine? And that starts with training and education, right? And that's another place that we have to ensure that everyone on staff at site is aware of what's moving forward. And then how do you actually lessen that burden? So a lot of these sites are large academic centers in the US and the EU, and they're usually specializing in that first in human and really working the beginning stages of a trial. But as as patients are treated, we have to think about the duration of these trials. And up to 15 years can be a long time. There is the expectation that these sites are going to have a lot of work in front of them. So we have to be cognitive of that and how we can ease that burden. And then we talked about this a little bit before with the product journey. Logistics, it can be extremely complicated, and it's very time sensitive. There's various things that we can kind of look at from an integration role, but the most important pieces of this are the chain of custody, the chain of condition, and the chain of identity, because there isn't a lot of room for error when product is moving across the board or across the country, or I know some sponsors that are even debating about moving it across the pond from the EU to the US, which is another set of complexity. And then ultimately, again, the patient journey, sophisticated recruitment strategies. How are we utilizing strategies to ensure we have the right subject patients coming in, and then the pre-screening through long-term follow-up. What does that look like for the patient? And then ultimately, patient support, which can be from the site, from the family members, etc. So those 4 items are really important when you start looking to operationalize your trial. And the rest of the presenters here are going to start giving you some insight to each particular one. So I'm going to pass this over to Marita, who again is our EU regulatory landscape leader, and she's going to take us through what it looks like right now in the EU.
Thank you so much, Desmond. So we can go directly to that slide. Thank you. We talked about the European Clinical Trials Regulation that came into force 31st of January last year as an option. And then from 31st of January this year, it's a mandatory process as far as it comes to initial clinical trials. It has completely changed the application and authorization process for clinical trials in the 30 countries that it applies to. So that's all the EU countries plus Norway, Liechtenstein, and Iceland. And the thing with this is it's a regulation, and by that it comes to the legal force directly in all countries that it's applicable for. So that gives us a very harmonized process. So submission documents requirements for all these 30 countries are all the same. There are some minor tweaks between countries, but very minor. The timelines are completely the same. And we use one submission portal for all these countries. So we can even do one submission covering 30 countries if we would like to. And some of the aims to put this regulation in place was to foster innovation and research in this region, including to make Europe a more attractive region to perform clinical trials. And also foreseeing in the future where you have clinical trials that target smaller patient population to more easily have clinical trials spanning many countries. And that's actually one of my favorite parts of the regulation, that it's very easy to add on more countries if you need to. The new countries have very limited power to question the decision already made, and the active process is relatively fast as well. However, for cell and gene, most countries do require an additional submission outside the EU CTR application. So that can be additional information to the regulatory authority or another country authority, but that approval process runs in parallel. So we still need to do this extra submission outside EU CTR. And then the actual submission process, if going back to EU CTR, is divided in two parts. So part one is the regulatory authority part. There you have the IB, the protocol, the IMPD, all these typical regulatory authority submission documents. That is sort of the scientific review of the protocol. And then in the part two, you have the ethical committees part, so to say. So it's more an ethical review. So there you find a submission documents like informed consent form, patient recruitment, also site documents and draft country budgets. And then if we go over to the next slide, there is some timelines here. So for an initial trial application, we have, yeah, no worries. It's stipulated maximum. Are you showing Desmond or?
Sorry,
No worries. So we have 106 days maximum for an initial clinical trial application. That is with the plus, because there are some addition when sub steps of the process fall into a week and then it's pushed out to the next weekday. So I normally recommend to count around 150 days or 120 calendar days to be certain for the time frame to receive the approval. With that being said, the timelines are strict in that sense that it can not be longer, but it can be shorter, depending on how fast the authority is to assess the application. And what's sort of new in Europe, at least, is that we also have very strict timelines to respond to the queries. So in the validation phase, for example, we have 10 calendar days, and in the assessment phase, we have 12 calendar days. So if you're unlucky and have the queries coming out on a Friday, you will hit 2 weekends, right? So you will have very few working days to respond to any queries. For these 7 gene therapies, that normally falls under advanced therapy medicine products. So there the authorities are allowed to extend the timelines as well. So they can use 50 extra days for the purpose of consulting external expert, but that is only for the part 1 where you have the protocol. So if you are unlucky and they request this extension, you can have the 2 different parts that normally run perfectly in parallel, being out of sync. So you can end up with a part 2 sort of approval on an ISF that is not applicable anymore, because in the part 1 you had a query asking you to update the protocol, for example. So one way to not risk being out of sync would be to, for example, only submit part 1 to start with. And that is perfectly fine under EU CTR. It will require a bit more time before you have the full approval and can start the site. But on the other hand, you can normally submit a bit faster because you don't need to have the sites in place, you don't need to have the informed consent in place. It's a more slim dossier, so perhaps you can do the initial submission a bit faster, and by that eating up some of these extra days. So that is something you can at least consider. So. And the next slide will show a little bit about what TRICE EUCGR are applicable for. I will not read through the full slide, but it's about a medicinal product, it's about humans, it's about testing efficacy or safety. It should be not normal clinical practice. And then we also have a new concept, and that is when you have a marketing authorized product and you use it as it should be used and it's low and minimal burden, then you can have what is called a low intervention trial. And then for the long-term follow-up that you typically are required, how does that work under EUCGR? So the regulation is still applicable if you write a protocol that will set the start of the clinical study after the exposure of the investigator product is finished. So thereby you could have the option to have a separate protocol that potentially covering several previous trials for all these countries, or you can of course include such follow-up directly in the protocol to start with, so you have both of these options. And by that, the regulatory EUCGR part was completed from my end at this time. So
Thanks, Marita. I really appreciate it. And that kind of brings us to our second polling question. So I'll give this back to Samaya. And really the question is, what is the biggest challenge of a site in cell and gene therapy? And I'll leave that for her.
Thank you, Desmond. Yes, so that does bring us to our second and final poll question, audience members. So you should be seeing that poll questions on your screens now. And once again, you can just go ahead and click on the option that suits you best. And that poll question is, what is the biggest challenge of a site in CGT? And your options are IRB submissions, or patient retention, or that could be patient recruitment, or logistics, or volume of data entry. So go ahead and click on the option that suits you best. And once again, that question is, what is the biggest challenge of a site in CGT? And audience members, just a reminder, if you have questions for all of the speakers, you can keep sending them in using the questions tab in your control panel. I'll give you a couple more seconds, audience members, to vote here, and then we'll go ahead and see the results. Okay, so I see that most of you have voted. I'll go ahead and share the results now. So the results are as follows. 14% of audience members voted for the first option, that's IRB submissions. No audience members voted for the second option, patient retention. 21% of audience members voted for the third option, patient recruitment. 50%, the largest majority, voted for logistics. And then finally, 14% went to volume of data entry. Thank you everyone for participating. Back to you, Desmond.
Thanks, I appreciate it. Ooh. Let me just make sure. There we go. So that takes us to two parts that I'll go over really quickly, and that's, once you get through the regulatory phase, now you have to look at what kind of sites you're looking to bring onto your study, and then some things that we look at from a logistics standpoint and kind of outlining the complexities that you might see. What is a site strategy that really makes sense, and what do you have to take into account? And what we think into the right, you kind of see how it's almost a jigsaw puzzle, but it all has to kind of come together. But I think it's really important that you have sites at this stage of the game where they have previous experience in cell and gene therapy. Naive sites at this stage, I don't know if it makes sense because it's such a complex process and making sure that they're properly trained. But you're looking for sites that you've previously collaborated with, that you previously had active trials on and move forward. A lot of times they're specialized in hemo centers who are priority for JC or FACT accredited institutions. We talked about that a little bit before, and that the oncologists are primarily ahead of the game because they've been really in the space for a lot longer than other indications and therapeutic areas at this time. You ultimately, for CAR Ts, want to make sure that you have a cell and gene therapy expert on site, and this could be anything from the oncologist to the particular staff, et cetera. Early phase experience is also a big plus. It's very important that they've run these trials before. They understand when the patients are coming in, the scheduling, the cell structure, where things are going out, coming in, things of that nature. Then you also want to be sure that they have the recruitment potential. I saw that one of the things in the poll, like retention, wasn't something that came up big, but there is a need to ensure that you have the right site for recruitment, and that can get kind of difficult sometimes because as you start looking for patients to fit the profile with inclusion and exclusion criteria, you need to ensure that they have a high incidence and access to the right patient population. When you have that, you also want to complement it with some referral networks. Overall, then there's the alignment of the standard of care with the protocol design. It's a protocol that has to be beneficial for the patient, beneficial for the site, that makes sense, and then that have favorable startup timelines. I think what you see right now in a large percentage is that the large academics have the majority of the experience, and we all understand that the large academics timelines may not match what you're looking for from your corporate goals or just the goals of the first patient in. So, you have to start looking for sites that fit the specs that you're looking for, but at the same time could utilize central IRBs in the US specifically. And those are in countries, that makes sense. Now with the ECTR, it's kind of leveled the playing field in Europe, specifically in the countries, since they all get submitted at the same time. And I think that's something also to consider. So that's kind of where you start with the strategy for the right sites, and you build upon that. And then you kind of go into the logistics. If we're talking about an autologous, and this is a really quick kind of design that I like monster trucks a little bit. So I wanted to use the monster truck icon, to be fair. So I found a way to use it in this particular graph. But you have to understand and we talked about this earlier, the inventory, your manufacturing slots, what's your constraints. Then you have to think about verifying the availability to schedule the patient out, the collection, the shipment, the apheresis. Then you have your cell collections, your transits, your shippers, who's bringing the product to the particular manufacturing site. Is the manufacturing, what is the timeline that they need to modify the cells, and how does that incorporate into the protocol, and how does that incorporate to the therapies? Maybe there's a bridging therapy that might be required while the patient is awaiting the infusion. And then there's the receipt of raw material manufacturing, cryopreservation. LN2 shippers can be complicated, and then you have to really look for your couriers. The autologous process is probably the more difficult of all of them, because obviously as you apherese and take the cells from your patients and then move it across the board, it has to be a really orchestrated effort between the sponsor, the CRO, the logistics vendors, which could include some software. There's companies out there, there's manual tracking, there's various ways we can go about that. But ultimately, autologous still is considered a difficult challenge for sponsors and CROs and vendors alike. As we move into allogeneic, it gets a little bit simpler. You can see that what it really signifies is that there's healthy donor cells that were actually being genetically modified. A lot of people will consider this off the shelf, right? I think at some point where we all want to get to is that if there's a particular matching to HLA for an oncology trial, or there's some type of match that we see, that they can just go off the shelf, grab the particular modified cells and make it a little bit easier to infuse a patient. So there's a lot of benefit to the allogeneic procedures, but at the same time, if you're looking into the overall landscape, they're not as successful at this stage, or they're still working. It's still a newer kind of process, and the autologous still are the lead on the approved therapies. And that really takes us to what is the product pathway and what are the logistics. You have to really understand the complex supply chains, the end-to-end processes, and the scaling up for manufacturing. As you go from a first-in-human, cohort managed or one subject per month, eventually, as you get to a phase 3 trial, you're looking for mass manufacturing, and you need to ensure that as it moves into that particular stage, we are looking at how we're getting everything achieved in the logistical process, and we can consider that people have referred to it as vein to vein or end-to-end processes. But what is the overall requirements, the transportation, extremely fast turnarounds from everything from what could potentially just be an assay that needs to be run by the central lab to the overall delivery of product on a Thursday, draw, apheresis. Do you account for the weekends, and how do you account for the weekends? Do you need to ensure that your slots and your apheresis and your cells are being shipped out on Wednesdays and Thursdays to account for Friday delivery versus a Saturday or Sunday where there might be a constraint? Those are all things that you have to kind of manage when you're talking about logistics, and there's various ways you go about it. As noted before, there's companies, vendor partnerships that we work with that put out really good software where they can actually track everything from the shipping integration to the scheduling of the patient to the overall approvals if the patient needs to move forward and then be apheresed or infused. So there's a lot of different ways you can go about that. And that kind of takes me to all of that information has to be tracked somewhere, and then you run into what does that data look like? How much data is coming in the first couple of weeks, and then Francesca is going to take us through that.
Thank you, Desmond. So yes, in the next 2 slides, I will cover some highlights in data management, some considerations that we really need to think about while we set up the database and clean the data. So I will cover 2 topics. One is the unique data flow and proactive measures, and another topic I will cover is the long-term follow-up and how we deal with that. So what is so special about the data flow? In these type of studies, the data collection is really front-loaded. So data is really key from the beginning of your study, and we need to have data review tools in place from study start. There will be a large amount of data collected prior to infusion. These patients, they are treated. There's leukapheresis and there's leukodepletion chemotherapies. Patients need to be monitored from that moment onwards, and then even they're not yet treated with the compound, but you really have to monitor those patients for safety reasons. Another thing that is different is that there is a single infusion instead of what we have in other trials, multiple doses, treatments over a longer period of time, or in the other oncology type of trials, you have cycles even, and it's a really long period that you are treating those patients and following up. Here you have your pre-treatment data collection with large volumes of data. You have your infusion, and then you have to continue monitoring those patients proactively to determine the dosage and also to make decisions on study continuation. So it's really from the start of the study, you really have to be on top of the data. Something else is the data integration that is needed. Data comes from multiple sources, lots of external data, lab data is to be collected, and there's also the cell tracking, as what Desmond just mentioned. That can be handled through a vendor, but something that we are currently investigating and starting is making use of the RTSM or IWRS modules within our EDC system to help and to support the tracking of the medication or the product. Then in some gene therapy trials, you really don't have a comparator, so something you need to think about upfront when the drug development plan is created, or how you set up your trial is where do you need comparator data and where do you get it from. So you could use real world data data from the patient's health records, from patient registries that are out there, or data from the laboratory information management systems. And that data you need to, has to be compared with your data you collect for your own trial. So really need to be kind of standardized so that that can be compared in your analysis. And then, of course, those patients are treated and the study is completed, but it's very important. There's this regulatory requirement nowadays to follow those patients for a longer period of time. Cell and gene therapy trials are really new. The therapy is new, especially when the patient's genetic code is affected. We don't know what it will do in the longer term, so that's why there's this regulatory requirement to follow those patients. During the long-term follow-up, there will be limited amount of data collected, and the focus will be probably on survival, but also on safety data collection. So especially areas of special interests, serious adverse events, and new malignancies. So we need to think about how do we best collect those data. There could also be basket trials. The basket trials, you have your main studies, multiple main studies, and then they are combined into one long-term follow-up trial. Also there, for the drug development process, you really have to think about how do you handle that. So what are we collecting in our main studies, and can we easily feed it into the long-term follow-up? Standardize the data collection so it integrates easily into the long-term follow-up. Probably in your long-term follow-up, you need to have some baseline data to know, okay, what was the starting point of those patients, and then, okay, what happens during the study? So can you feed the data from your main study into, like multiple studies, into the long-term follow-up study? Those are things you really have to think about upfront when you start your main study. So how do we collect the data, and where do we collect the data? You could start, if it's just one main study and followed with a long-term follow-up, you could use your EDC system, same EDC system, continue collecting the data, but the question is if that is wise, because the data that you will collect is limited, and the uptime of your database will be very long, so it could be quite costly. Your maintenance needs to continue for that same EDC database, and maybe it's better to basically separate, close the main database, start a new database, and collect your long-term follow-up in that database. Could also make use of decentralized trial platforms that would help patient engagement, but then you could also use some of the modules in the platform, like the ECOA, EPROA, the EPRO, to collect the patient reported data. So you can ask patients and follow up with them, like, how are you doing? You can set notifications and get their ask them about any events that happened during the time. What we also are looking into if you use EPRO for the long-term follow-up is to use a standard pick list so that it is standardized, that the data that is collected from patient directly is standardized. And the other alternative, because there's a special interest for AEs, special interest, malignancies, is to ask patients to enter the data in an ECOA device or in EPRO, and then have the investigator or the doctor assess what is relevant to mention in an EDC. So patients don't really know what is really relevant to mention. They can say anything, headache, whatever, but that may not be the thing that is of interest in this long-term follow-up study. So then the doctor can question based on what the patient entered in the diary and enter the relevant information in the EDC, and then you can code and use that for the analysis. So what is the best option? It really depends on several items. It depends on the duration of the trial, of the long-term follow-up. It depends on the volume of the data that needs to be collected, the visit frequency within the protocols, and also who enters the data. Is it the patient or is it the site? So yeah, lots of things to consider, and then I would like to hand over back to Desmond.
I know we're approaching the end of the hour, so I want to ensure that Joyce has appropriate time. So Joyce, please take us through the patient engagement strategies.
Yeah, we can move to the next slide. Thanks, Fred. So just going to start off with patient engagement and sort of talk about kind of the past experience and the current experience that we've had working with cell and gene therapy. So really today, like my experience has been within kind of small patient populations, a lot of oncology studies, some rare disease studies where the patient population is very small. And actually, my experience today is that the recruitment isn't the major issue, actually, because the trials are actually very compelling for this patient population. I think recruitment is going to start to become more of an issue, and we need to have strategies to engage with that as we expand into larger patient populations. But all that being said, so Des did mention, obviously you're going to have a couple of academic sites, and you do need to be able to have referral networks between all those different oncologists and rare disease experts. And that's something that we would just like to set up and making sure that those referral networks were working. But actually take it back to the basics. We're asking a lot of these patients. There is a big choice that they have to make. And so just simple information to make sure that we are explaining exactly what's going to happen, when it's going to happen, understanding the process, understanding the timeline, both for the actual maintenance, for the treatment, and just, and then on to the commitment for long-term follow-up. And that has to be in material that is taken into health literacy, so it's simple and understandable. We do put, within clinical trials, we put a lot of expectations on our site to explain studies and explain what's required in a study for a patient. But there is, I like to kind of remind myself that there's studies out there that are showing sites often believe that they've explained very adequately to patients, but there's a particular study where 80% of oncologists got it. They had explained the diagnosis and treatment options, but 80% of those same patients didn't understand actually what's been described for them. So it's one of the things I think is absolutely essential is to make sure that patients actually have the information in their hands that they can take home, discuss with their families, and making sure that they actually are understanding the whole journey of the way through. We did talk about, as Gal mentioned, long-term follow-up and the use of DCT platforms. We actually have to make sure that patients are aware of what the expectations are there as well. But what we're seeing within cell and gene therapy is the market starting to move beyond these smaller patient populations, and we're moving into more prevalent diseases just as we get some more information around the genetic impact of different disorders. And so actually we're starting to see that we're using more sophisticated methods to engage and recruit patients. It may well be now that to take the people who the specialists may no longer be the oncologists, but the oncologist on the site that actually has a speciality in the actual technical aspect of cell and gene therapy. So we might have might need to make sure that those two different specialities are actually talking to one another, that they understand one another, and also that both of them are describing to the patient population exactly what's going to happen. We're starting to look at EHR data as a way of interrogation to identify patients, patient communities, and patient advocacy groups. are all coming into the mix when we're going beyond that small patient population. So actually coming more like a lot of other clinical trials and what we're doing in order to engage the patients to start with, and then to recruit the patients to start with, identify, recruit, and then keep them engaged. So move on to the next slide. Really just this is a quick, just to say that when we're talking about the long-term follow-up, so actually once we've got the patients, they've been involved in the actual the treatment, depending on what the vector is, obviously, depends on how long they're going to need to be followed up by for regulatory purposes. The site and the sponsors may change during that if it's a 15-year follow-up, but actually importantly, the patients and families, they may not be, they may be okay. There's obviously many of the cell and gene therapies are targeting children, and so that child during the time of 15 years moves from being a child to an adult. And we still want to make sure that we're actually following the child, following the patients, and making it easy for them to continue that engagement, because it's obviously important for the pharma companies, important for the community as a whole to understand all the risks associated with cell and gene therapies. It will make it better in the long run. So moving on, just to talk about some of the solutions that we have at Allucents. I want to change next slide, sorry. Before we do, what we like to do with most of our our strategies is actually is listening to patients. As part of a larger patient voice exercise in AML patients, we were asking them about their options that they would like to follow for long-term follow-up. So we asked them about taking, if they were involved in a 15-year long-term follow-up, we gave some of the parameters to that and asked them if they would be happy with some of the assessments being done outside of the traditional site setting. There's all various quotes there, but really what we heard from the patients was that having remote assessments actually would make a big impact for them. All the patients we spoke to did talk about the financial burden of travel. They talked about the logistics burden of travel, and actually just like how difficult it is for them dealing with the disease and having to do the travel. One interesting thing was a number of the patients did say that they needed to have some they had some interactions, direct face-to-face interactions with the sites, because they needed to have that built, that trust built. So it's interesting that patients have actually seen that, they can see that forward. If we just go to the next slide, we have a schema of what we are looking at as our sort of standard long-term follow-up. So as we said, we have a decentralized platform we work with. We work in partnership with Thread, and we have worked on what this looks like in a very collaborative manner. It's depending on whether it's a 5-year follow-up or a 15-year follow-up. Regulatory requirements for on the whole after the 5-year period is a yearly touch point with the patients. What we are seeing is actually some of our cell and gene therapy long-term follow-ups are actually collecting more data than just the survival and the malignancy. And Francisca kind of mentioned that there's many of them that are collecting patient-reported outcomes. There are some of the trials that we're seeing coming through that are also collecting various assessments which do require the patients to actually go onto site. But we have a methodology where we can engage with the patients over that 15-year period, where the majority of the visits can be remote. What we are suggesting and what we're doing is, even though the regulatory requirements are only touch and base yearly, it's actually having a little kind of touch in with the patients on a quarterly basis, just to make sure that they're still engaged with us. And if they don't reply, then that would be a trigger for our site to get in contact with the patient and make sure that they can follow up with them rather than waiting till you get to the 1 year period. And having a decentralized platform also fits in very well with basket studies, so you can have that as a single database. Obviously, that all needs to be planned up front. There needs to be a lot of thought in. The data collection might be different from each of the patient populations. The timing would need to be the same. Having a basket study doesn't really affect patients as such, but it could be beneficial to the sponsor. It could be beneficial to our data management colleagues, and it's actually obviously a little bit cheaper in order to operationalize. So we do look at it from patient preference, site support, the regulatory environment, and just really having kind of laser focused on ensuring that the patient burden is as low as possible and allowing patients to get on with their life. And then if we just go to the next study, the next slide. And this is really just a wrap-up slide to say that from end to end, patients should be engaged in the full process. I didn't talk very much about study design, but that's absolutely essential that patients are involved in the study design, and that they're involved in the recruitment. We do actually do for some of our studies like co-development of materials with patients. We engage PAGs and patient communities. It's obviously very important. And then moving on to treatment, it's obviously very important to keep patients informed. Any travel, accommodation, support needs to be included, and then into that long-term follow-up, trying to move more of that into a remote, away from traditional sites, just allowing patients' families to get on with their life and not having to think of themselves actually as patients. So with that, I'll wrap up. I think we've got a couple of minutes, just thank you.
Thanks, Joyce. And I just want to thank everyone for the time today, and I'll pass this back to Samaya.
Well, thank you very much for that insightful presentation. Yes, so that does bring us to our Q&A portion of this webinar, audience members. So if you have more questions, keep sending them in. We've already received some questions, so let's start off with that first. And our first question here is, do you recommend manual tracking of logistics or vendor managed? What are the options?
I'm sorry, Samaya, what was the question?
So that question is, do you recommend manual tracking of logistics or vendor managed? What are the options?
So it's a great question, and my preference is always to use a technology that takes away the human error. I think manual works well in first in human because it's a very limited number of subjects. What we really need to start thinking about is how do you go from the manual tracking in your phase 1 first in human, where you can kind of support that, but then how do you grow that into phase 2, and now you're looking at maybe 20 additional subjects, additional countries, and then eventually when you get to phase 3, it's pretty much unmanageable at that stage. There's various ways we can go about that. So there's vendors, one of the vendors of such is Traxel, and they have 3 different types of platforms that we partner with Traxel, and we work everything from, you know, their first in human, where they have like a light program, and then they have their heavy duty where there's a ton of integration. My preference is to always start early, although there is some and there is some costs to that, and I know that's probably the one thing that always gets measured is, do we need it now or do we need it later? But if you set yourself up early on and you know, take the manual out of the process, you're going to have a better structure as you move into the advanced stages. So that's normally my preference on how to go. And then there's also the potential, and depending on your platform, and maybe Francesca talk a little bit about this, but basically, you know, reconfiguring systems within the EDC is also a way that you can go through this.
Thank you, Desmond. And we have another Very interesting question from an audience member. So the question is, I have two questions around site selection. In the presentation, you suggest using sites you have worked with before. This isn't always feasible in rare disease and becomes more complex when you are pursuing CGT in a rare disease space. Often, the indication really requires utilizing sites that are not familiar and or have not previously conducted CGT. Secondly, use of repetitive sites doesn't really support the initiative of achieving patient centricity. How do you overcome these two considerations and still maintain the speed of bringing up a site to activation and diverse patient enrollment opportunities?
Great question, and honest, and to be fair, as I was reading it on the side, I also saw how it was answered in the question, and I agree with the premise. So I think it is a challenge, and I think, you know, the first part of the question is, you know, when we recommend sites that we've previously used, it's most times in regards because we're confident that, you know, at stage they can potentially meet the demands of what you're looking for from and it's always hard to kind of, when you run a clinical trial, we obviously understand that what we're looking for are the new cures in patients. But at the same time, there's individuals, you know, companies that are, you know, dealing with their Series A or their Series B, and they're just looking for additional funding to move to the next stage. They need success, and they also need to meet certain requirements. So it's a delicate balance. So, you know, when we recommend sites that have been previously used, it's mostly to meet the expectation of maybe what the corporate goal is. And that's based on the fact that they have that experience. But in rare disease, you're absolutely right. You know, there's more and more you have to start working with the advocacy groups and really work to tie them in, to bring in the investigators and PIs that may not be acutely involved in CGT, but then it's up to the sponsor and the CROs and to you know, bring them up to speed, train them. And one thing that you have to start looking at, and this is something that, you know, you see a little bit here or there, is what if they don't have an apheresis unit? You know, a site that's maybe in a rare disease or some type of you know, autoimmune disease. Do you have the ability to, you know, start looking at like these secondary sites where they're doing the apheresis and tying them and getting them where the experience of that is taking place for a PI who may not have that experience? So it's a great question, and it's, you know, I don't know, Joyce, if you want to add a little bit about the advocacy group, and then I'll take the second part of that as well, because, you know, I think the other part is the diversity, or Joyce, maybe touch on both of them and close it up.
Yeah, and we are starting to see sponsors speaking to us in smaller studies as well about diversity and inclusion plans. And so that's something that we can build in. So we can build that in at the study level, at the country level, and then track it with our site. It's also about, you know, I guess I alluded to, you know, in the past, there were, it was mainly within the oncology area, and maybe the sites themselves were actually recruiting the patients directly or from their own network. I just think that's becoming more complex now, and so we need to make sure that the recruitment strategies that we're using are actually engaging with all the patient population that is needed to fulfill that original diversity and inclusion plan that we put in place. So that's something that is definitely becoming more prominent. We see it in other types of indications, but it is coming over to the cell and gene therapy area. So it is always that sort of thing between going with your trusted partners, but then expanding into other areas. We have looked at some networks, site networks who actually have more dealings with with kind of community centres in the US, and then into the long-term follow-up, which is a slightly different question, but something that we are looking at virtual sites there as well to make it actually easier for patients to continue to be involved. So there's a variety of things that are going on, but yeah, diversity and inclusion is definitely going to become something that's prominent here, and that's all around our recruitment plan.
Great. Thank you so much for all of those answers. So we reached the end of the Q&A portion of this webinar, but not to worry, audience members, if you do have more questions, you can keep sending them in using this again. So just take this down. That's info@allucent.com. So if more questions, more comments, you can, of course, use that. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as will help us improve our webinars. Now, please join us in giving a big thank you to our speakers today. So that's Desmond Cabrera, Joyce Moore, Marita Krushkoff-Osterberg, and Francisca Sampson. So thank you, everyone. Hope you have a fantastic day.
Thank
Thank
you.
you.